Good afternoon. So we're just after 12 o'clock, and I know a lot of participants are still joining, but out of respect for everyone's time today, I think we'll get started. Welcome to Uncover Your Soil Health, and thanks for joining us today, and happy World Soil Day. My name is Madeline Rodrigue, and I'm the Knowledge Mobilization Specialist at the Ontario Soil and Crop Improvement Association. Today's webinar is offered in collaboration with the Greenbelt Foundation, Soils at Guelph, and the Soil Health Institute. Before we begin our program today, we would like to recognize the long and rich history of First Nations, Inuit, and Métis people in the place we call Ontario and show respect for their traditional territory on which we live, work, and farm. Today's panelists are going to introduce two exciting soil health initiatives in Ontario: the Soil Health Assessment and Plan Ground Truthing Project and the Greenbelt Soil Health Testing Program. You will learn about what they're doing, what's been learned so far, and how you can contribute while also learning something about your own soil health. We'll reserve 10 minutes at the end for questions, and as you think of questions, we encourage you to add them to the question and answer box below. Both of these programs that you'll hear about today were made possible by funding and support from the Weston Family Foundation and the Government of Ontario. Our first speaker, who will be talking about the SHAP Ground Truthing Project, is Inderjot Chahal, the postdoc research fellow at the University of Guelph.
Madeline. So welcome everyone to my presentation. So for today's talk, I will be discussing about the Soil Health Assessment Framework for Ontario, what it is, and what are the soil health indicators that are being included and analyzed as a part of this assessment, and how can we build on the existing SHAP in terms of capacity and database assembly. So let's background about the Soil Health Assessment Plan, also called as SHAP. So this was initially developed by OMAFRA. So it is a mobile or a web app-based tool that was designed to help the growers in understanding and identifying the soil health challenges and what management practices they can undertake to address those challenges. So it's a very useful and valuable resource that enables the users to create soil health baseline or help them to monitor their soil health over time, and also is great for comparing different fields or different sections of fields to one another. So there was a SHAP guidebook that was released by OMAFRA. So it's a very valuable and comprehensive resource. It highlights and mentions the details about this tool and the indicators that are included as a part of SHAP assessment, and also gives valuable advice in terms of data interpretation related to those soil health indicators. So here on this slide, you can see the link for that guidebook that was released by OMAFRA. So as I said, SHAP is a very valuable resource. It gives you an overall estimate or assessment of your soil health. So it includes physical and biological soil health indicators, which is a great resource because it complements your soil fertility test, which essentially includes soil chemical indicators. So overall, you get a good package in terms of physical, chemical, and biological health of your soil. So the indicators which are included in the lab analysis for the SHAP project are soil organic matter, active carbon, soil respiration, potentially mineralizable nitrogen, and aggregate stability. So it is a combination of physical and biological soil health indicators. Now, when we talk about these indicators, how it works is usually it's the lab will give you the results, and then these data, the data that you get back from the result is converted to a soil health score for each of these indicators, and then you can get an overall soil health score based on these scoring functions. So that's how it usually works, and that score determines how good, bad, or moderate your soil health is. So let's talk a little bit more about the soil health score. So soil health scoring, it is it's very complicated. It is challenging assessing and interpreting those soil health scores. It is very challenging because there is a complex interaction of these soil health indicators with the inherent soil characteristics, with the land management practices that a grower is adopting at their farm, also with the environmental conditions, and of course with the location where the data or the sample was collected. So out of these different variables that are influencing the soil health scores, the inherent soil characteristics is a major one. So soil texture is one of the main attributes that causing the variation or the variability in terms of assessing your soil health scores. So therefore, we need to develop a regional based soil health scoring functions that will help us to accurately assess and interpret our soil health data. So when we talk about these scoring functions, which are used to develop these soil health scores, so there are main three types of. First one is more is better. So here you can see essentially indicates a high measured value implies a high score. So more the value of the soil health indicator, the more will be the score associated with it, and the better your soil health will be. For most of the soil health indicators, this scoring function is commonly used to score the soil health indicators. Is the less is better. So here, high measured values indicate a low score at poor soil functioning. So for instance, for some of the micronutrients such as manganese and iron, we mostly follow this less is better approach in terms of scoring them, because sometimes excess concentrations of these nutrients in your soil can lead to toxicity. So we follow the less is better approach for scoring these soil health indicators. Or the scoring function is the optimum curve. So here you see it has an optimum range. Scores are low when the measured value falls either below or above a certain range. The most commonly used indicators that are used that use optimum curve for scoring the soil health are extractable phosphorus and pH. So for those indicators, we mostly use the optimum curve to score them. So these are the commonly used soil health scoring functions when we develop these soil health scores. Talk about the indicators that are used in the SHAP, why these indicators are important, and how they are interpreted in terms of scoring functions. So first one is the soil organic matter. So soil organic matter, as we all know, is one of the most important soil health indicators. It usually results in better soil health. It helps in developing resilient soil systems. Also is a great source for increasing the soil microbial populations and their activity. It also acts as a source of food to soil microorganisms. So if your organic matter is protected within the soil, it can also act as a long-term sink of carbon. But if it is exposed, it can also act as a slow release pool of nutrients to the soil. So in terms of interpretation, so usually we follow the more is better scoring function for interpreting soil organic matter. So more soil organic matter usually reflects a better soil health and better soil functioning. The SHAP is active carbon. So active carbon, it represents a fraction of soil organic carbon. It's about 1 to 4%. It is considered to be relatively labile, and it serves as a readily available source of food and energy for the soil microorganisms. So it responds relatively quickly to the change in land use management than the soil organic carbon. For this indicator also, we follow the more is better scoring function in terms of assessing the data. Other indicator included in SHAP is the soil respiration. So soil respiration is an indication of a potential mineralization of soil carbon. So it indicates the soil biological activity. So in this case, so usually when we have high respiration rate, it indicates more biological activity. So we follow more is better approach as well. But here we have to be a little bit more careful in terms of assessing it, because sometimes very high respiration rate also leads to relatively faster and excessive mineralization of soil organic matter. So in that case, we have to be really careful in terms of how we want to approach this indicator in terms of scoring for the soil health assessments. Indicator is the potentially mineralizable nitrogen. It measures how much nitrogen is present in organic matter that would be converted to the plant available ammonium over time. So it indicates the amount of nitrogen that is, or indicates the nitrogen supplying capacity of the soil. So here it is also used in terms of Morris-Bader approach is used in terms of assessing it, but it also indicates, but high values of this indicates also that there is a loss of nitrogen during the non-crop growing seasons and also during the wet conditions. So again, like soil respiration, we have to be really careful with the potentially mineralizable nitrogen when we are talking about scoring these indicators. The indicator, the last indicator that is included in SHAP is the aggregate stability. It is a soil physical indicator and it measures the resistance of soil aggregates to disintegration from disturbance due to tillage, raindrops, or from the water erosion. So results of this indicator are usually expressed as the percentage of stable macro aggregates. Again, for this one, we usually follow the Morris-Bader approach for indication of increased soil health. These were the indicators that are included in SHAP, but now let's talk about what SHAP exactly is and how it works. Okay, so we know now about the indicators that are included in the SHAP, how we can get more information about SHAP from the guidebook that's released by OMAFRA. So this tool consists of two parts. The first one is the soil management survey, and the second is the in-field data collection. So what happens is when a submitter or a grower is interested to be a part of this project, so they'll complete a SHAP management survey. Once that survey is completed, you'll get an email that will indicate your survey ID, and we'll have another link that will give you access to the in-field data collection form. So once that form is completed, then the users usually get a SHAP report template that they can use to fill in the soil health data that they receive from the lab. So it's a fairly straightforward process to get an easy interpretation of your soil health. Now, the next component of my talk is about the ground truthing project. So this was about the SHAP that was developed by OMAFRA. So now let's talk about the ground truthing project. So this is a pilot project. This is still in early development. So this project was funded by the Weston Family Foundation. So in this study, the University of Guelph researchers, they are working closely with OMAFRA staff in terms of creating and populating an Ontario-wide soil health database. So the goal is to develop a soil health database that specifically for Ontario, and it will be covering a wide range of Ontario soils. The other major goal of this project is to develop scoring functions that will be used to compare individual soil samples to other soils, and also to refine the scoring functions that are already existing and how we can refine them based on the soil texture, because now we'll have more data. So that would be a really valuable source in terms of developing and refining those scoring functions. So what is included in this project is we have a free SHAP analysis that includes all these indicators that we just discussed. So you get assessments of organic matter, aggregate stability, active carbon, potentially mineralizable nitrogen, soil respiration, and aggregate stability. So for the soil, we also get soil texture analysis in terms of estimates of percentage of sand, silt, and clay. There's also free soil fertility analysis where you get all the assessments of your nutrients, such as phosphorus, potassium, calcium, and magnesium, and pH and organic matter. So those are the things which are included in this ground truthing project. So the next question is how project and what are the steps that a person should take to be a part of this project. So as SHAP, it is also fairly straightforward. So there are numerous steps that you need to take. So the first step is if you're interested and you want to participate in this project. So the first step is you have to send an email to the Soils at Guelph. And in your email, you need to highlight your name, county, how many agricultural fields you're planning to sample, how many samples you're planning to collect from each field, those type of things. Once that is completed, then you'll get an email that will have a link to complete the soil management survey. So once, and it will also have a link, not a link, it will also give you an information about your unique sample ID. And in that email, Once you complete that, the soil management survey is completed, you'll get another email that will have a link to complete the in-field data collection form. So you'll access that link, complete the in-field data collection form, submit that form, and once that is completed, then you can go out and collect the soil samples. So in terms of soil sampling, we usually ask the submitters to collect about 20 to 30 soil cores up to 6-inch depth increments, 6-inch depth in the soil. And once that sample is collected, you mix it, homogenize it, and then you pack it in a soil sample bag, and 2 boxes from each field we ask for. And then you can pack them up, label the bags with their sample IDs. The samples that we are collecting are analyzed by the Agricultural Food Laboratory at the University of Guelph. So they have their own submission form. So once you collect your samples, you have to fill in a submission form from that lab. So you also need to include that submission form when you're shipping your samples to the lab. So that's a whole process of sending and submitting your samples to the Agricultural and Food Laboratory at the University of Guelph. So once the samples are submitted to the lab will undergo and undertake the soil health analyses on those samples, and you'll get a report on your soil health back directly from the lab. So you'll have your email ID, and the lab will send back the results directly to that email ID. So for this year, so this project, the ground truthing project, started in 2023. So we had about 250 samples collected so far, and we've been working closely with the Agriculture and Food Laboratory, who is analyzing our soil health samples for this project. So we got some of our results back from them, not all. So we have about 84 data points so far that we have received back from the lab from this year, so from 2023. So I'm going to present some of our preliminary results in terms of the management variables that we are collecting as a part of this project. So the first variable is the tillage intensity. So under tillage intensity, we have 4 subclasses. So we have light disturbance, moderate disturbance, no-till, and strip tillage. So we found that there were statistical differences amongst these tillage categories for the active carbon and soil respiration. So for active carbon, as you can see here, strip tillage had the greatest active carbon, whereas light disturbance and moderate disturbance were the least. Now, this is not our full data set. I would like to re-emphasize this. This is only from 84 data points that we have received so far from the lab. So we are still in the process of getting more data from the lab as it's being analyzed. So for the soil respiration, we also found no tillage had the greatest soil respiration, whereas moderate disturbance had the least. We did not find any statistical differences amongst these tillage intensity categories for soil organic matter and the potentially mineralizable nitrogen. Interested in is the usage of cover crops or the adoption of cover crops in the cropping systems. So we had the categories where there were cover crops in the cropping systems and versus no inclusion of cover crops. So contrary to what we expected, we did not find any statistical differences between these categories for all of the soil health indicators that for the data that we have received so far from the lab. There are some numerical differences, but we are not able to pick out some statistical differences as far as now. The variable that we are interested in is the cropping system. So we have 2 subcategories under this variable. So first one is annual grains and the other is annual grains with forages. So we except active carbon, we found some statistical differences between these categories for soil organic matter, potentially mineralizable nitrogen, and respiration. So we did see significant increases in organic matter, nitrogen, and respiration when we have the forages in the cropping systems versus the annual grains. There were no statistical differences between these categories for active carbon. And this result was quite expected, like in terms of greater soil health and soil health parameters when you have forages in your crop rotations versus the grains. So this is the data that we have so far from those 84 samples. But it will be interesting when we have more data analyzed and getting back from the lab. So once we have a complete data set, it will be interesting to see how these trends change and we might be able to tell a better story once we have a full data set. Let's talk about the next steps. So right now, as I said, we have our target for 2023 were to have at least 250 samples that we have achieved, and we are on our track for that. So currently we are recruiting samples for next year. So our plan is to have at least 500 agricultural fields in 2024 and 250 in 2025. So our preference right now is more on the light textured soils and also in the Northern Ontario soils. We are focusing only on getting samples from agricultural fields, and we are not sampling and collecting data so far from organic and muck soils. So our preference is mostly agricultural fields, light textured soils, and Northern Ontario soils. So as I said, it's a pilot project. So this SHAP ground truthing project is a pilot project. It is it's still under development, and it is a slow process. So we need to manage the expectations of the people who are currently, as well as currently enrolled in this project, and also who will be enrolling here in the future. The analysis of the soil health indicators by the lab, it takes time. It is pretty labor intensive. The analysis itself is very intensive, so it takes time. So that's why it's a relatively slow process. But we are working closely with the lab, and we are trying to get as much data as possible, as quickly as possible, so we can have some good soil health data and be able to interpret it in a way that is easy for users to understand. So for now, the current resource that we have available in terms of assessing and interpreting the soil health data that the users are getting directly back from the lab is that guidebook that I mentioned in the beginning. So that's the soil health assessment plan guidebook that was released by OMAFRA. It is a great source for now in terms of interpreting your soil health data that you're getting back from the lab. But also our goal for next year is to work closely with OMAFRA in terms of developing a soil health report template that will be more user-friendly, and it will be helpful for the users in terms of easy interpretation of the results that they're getting back from the lab. So that's our goal for next year as well. So those are some of the next steps that I wanted to talk about today, and in terms of recruiting more samples next year, and also how we're building in terms of database and capacity for this project. I would like to acknowledge our funding source, who is the Weston Family Soil Health Initiative, also the Agriculture and Food Lab at University of Guelph for analyzing all of our SHAP samples. And if you have any questions related to this project, or and you want to enroll in this project or participate for the next year, please feel free to email me. My email ID is listed here. Also, we have the Soils at Guelph account that is also monitoring our emails as people are contacting us and are showing interest to be a part of this ground truthing SHAP project. So with this, I would like to end my talk, and maybe you'll have some couple of questions that we will be taking in the last section of our presentation here on the webinar. Yeah, thank you.
Thank you. That was a lot of information to cover in a short period of time. So I'm sure there's going to be a number of questions to get to at the end. We're going to move on to the Greenbelt Soil Health Testing Program. I think you'll find that these programs complement each other quite a bit. And so I think a lot of the discussion towards the end will flow really well for both programs. So to introduce that program is Jenny Bauer, Research Soil Scientist at the Soil Health Institute, followed by Ryan Carlo, Soil Health Specialist at the Greenbelt Foundation.
All right, happy World Soil Day. It's great to be here with you all. I'm Jenny Bauer, a research soil scientist with the Soil Health Institute, and I want to talk to you today about benchmarking soil health in the Greenbelt. So how do we define soil health and how do we quantify it? One definition is the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. And so we need to select indicators of soil functions, which Inderjot presented some wonderful information on. And then we also need to interpret indicators in the values in the right context, right? So for example, an organic matter percentage might be, or value might be low for a sandy soil, or sorry, might be relatively high for a sandy soil, but low for a clayey soil. To address this, the Soil Health Institute has developed a benchmarking approach, and this is an interpretable, scalable, and locally relevant method for evaluating and monitoring soil health. I want to walk you through this figure because it's important. So we've got this line representing different soil health indicator values. So, say it's organic matter. We've got low indicator values on the left and high values on the right. And you'll notice these 3 brackets. So basically, when we go out sampling for soil health, we go to a specific region that has, similar parent material and climate, and we select from 3 different managements. So we're gearing our sampling toward managements that reflect the low end of soil health indicators, such as annual high-intensity tillage, and then soil samples that reflect incorporation of soil health practices like minimum till, no-till, and incorporation of cover crops. And then we also sample soils on reference sites. So these include fence rows, pasture, long-term undisturbed perennial sites. And the point of this is to give a sense of the ranges of different indicator values for management systems so that a producer can can receive a report of where their soil is at. So where is your soil health value on this spectrum? And then where might you see it going if you were to incorporate some of the beneficial management practices that we know can increase your soil health? An important thing here, though, is that we are not in the business of setting goals. That's done by farmers in collaboration with their advisors. To study this in the Greenbelt, we partnered with the Greenbelt Foundation on a 4-year partnership. And as mentioned, this is funded and supported by the Weston Family Foundation Soil Health Initiative and the Government of Ontario. So our project measures soil health on grain and oilseed farms in the Golden Horseshoe. And we sampled 124 sites this year. And that's what I'm going to present data from. Ryan will talk more about our next steps. So as I mentioned, we grouped our sites by actually their soil properties and also by their management systems. So the management systems we were looking at included baseline sites. So these might be in continuous soy or corn with annual high frequency tillage. We also sampled soil health management systems, which I'll abbreviate as SHMS, which might have incorporated winter wheat and some cover crops and were minimum till or no till. And then we sampled some of these reference sites, which were included prairies and fence rows and long-term pasture lands. There are a lot of measurements on this slide, but luckily Inderjot went through some of them, so that is great. But the thing you need to know, so we were kind of merging Soil Health Institute's recommended indicators with the SHAP test, and there's a lot of overlap. So so both of us are analyzing respiration and aggregate stability, and yet I'm going to focus on a subset of these, and I'm going to focus on soil organic carbon, which you can think of as really similar to organic matter. So these images show a vertisol, one that has had all of its organic matter depleted, and then another that still retains a lot of organic matter, which gives it that nice dark color, as well as the structure. So organic carbon is around half of organic matter. And so it's a good way of representing that, but a little more measurement specific. We also measured aggregate stability, which like Inderjot mentioned is the capacity of these little pea-sized aggregates that you can see in this image to resist falling apart when they're submerged in water or when a rain event occurs. And then the third indicator I want to look at today is potentially mineralizable carbon, but let's just call it respiration. So this is a way of quantifying microbial activity. I want to take a brief commercial break. So the second indicator that I mentioned, aggregate stability, is something that you can directly measure on your smartphone. So it's World Soils Day, and the Soil Health Institute launched this app called Slakes, and I encourage you to seek it out. We'll be sending a link to the app in the follow-up materials from this webinar, but you can also Google it. So I encourage you to download it and try it out for yourself. Okay, commercial break over. So for our first year, as I mentioned, we sampled 124 sites, and we sampled these in June and July from the shaded area. So we went to Peel, Halton, and Hamilton, and we sampled soils on around 60, working with 61 land managers. This is a texture triangle showing the distribution of soil textures that we sampled. So these are real texture analyses. And what I want you to take away from this is that we sampled a range of sand and clay values. So they formed a nice sort of continuous line on our texture triangle. Again, we had a relatively small region, and that means that we can model the influence of clay and sand continuously within our data. We stuck to mostly well-drained and intermediately drained soils, as those represent the dominant drainage classes under cropland. So in terms of our grouping by management, we ended up sampling around 38 baseline sites, 31 soil health sites, and 48 reference sites, leaving out 7 that did not fit in these categories. So 31% of the sites with soil health practices were no-till, the rest were minimum till, and 71% of the sites with soil health practices had at least one cover crop in the past 5 years. The soil textures we sampled included sandy loams, silt loams, silty clay loams, and clay loams. And the mean annual temperature range was around 6.4 to 9.1 Celsius, and precipitation from 830 to 950 millimeters. All right, let's get into the data. So I want to walk you through this graph because you'll be seeing some similar ones in the next few slides as well. So I modeled the influence of a bunch of different variables on our soil health indicators. And what I'm going to be showing you is the factors that were most important to explaining the range of values that we observed. So in the case of organic carbon, which again represents around half of organic matter, so you can think of it that way, I'm giving organic carbon on the y-axis from values from around 1 to 3.5. So you can think of that from around 2 to 7% organic matter. But notice that the values plotted on these lines increase with clay content, which is on the x-axis. We sampled from around 10 to 30% clay. So what's important here is we found that management was important, and so was clay, as we might expect, right? Texture and some of these management practices. And we found significant differences between our baseline, our soil health sites, and our reference sites. And this explained around half of the variation in our data. So we got an R-squared of 0.47. We found that sites with soil health practices had 1.2 times higher organic carbon than baseline sites. And then when we looked at reference sites, these had 1.8 times higher organic carbon than baseline sites. Something to notice here, though, is that the magnitude of that difference, even though it's a ratio, changes as you increase the clay content. And similarly, what would be considered high or low in this system changes based on the clay content. So at 10% clay, if we have a value of like 1.6 for organic carbon, that would put us on the soil health curve, right? But actually, once we get to 20% clay, that becomes more of a baseline value. And the important thing here is that context matters, right? Our soil texture helps tell us how much organic carbon we can expect and how much change we can expect from management. When we looked at aggregate stability, instead of clay being a dominant texture factor, we found that sand was more important. So here we found that actually aggregate stability increased with increasing sand. And that might seem a little counterintuitive because sand doesn't aggregate very well, right? But it turns out when it does aggregate, it actually, it slakes a little bit less. So it's not shedding those, particles in the same way that clay does. So we found that, again, there was a significant difference between baseline and soil health sites, and that soil health practices had 1.2 times higher aggregate stability. Again, I know it's weird that the R-squared is the same, 0.47. I swear if I gave you the full numbers, they would be different. But it once again, our model of sand and management explained around half of the variation in these values. And when we looked at reference sites, these had 1.4 times higher aggregate stability than baseline practices. So I think an important thing here is that we're seeing that soil health management, so adoption of reduced tillage and incorporation of cover crops, is changing our soil health indicator values, and there's still room to grow in that regard. I want to bring you to potentially mineralizable carbon. So unlike aggregate stability and organic carbon, we didn't find that texture was important for this indicator. However, management was really critical in explaining variation in this soil health indicator. So including management in our model gave us an R-squared of around 0.65. So explaining nearly 2/3 of the variation in this indicator. We found that sites with soil health practices had 1.2 times higher respiration than baseline sites. But reference sites had 2.3 times higher potentially mineralizable carbon. So that was a lot of data, but I want to show how we actually convert the modeled influence of these variables into farmer reports. So what we do is for a producer's specific soil texture, say we have a clay content of around 17%, we can draw a line on that organic carbon plot that I showed earlier. And we can also add our confidence intervals. So I didn't include the variation around the mean in the previous plots to kind of simplify the story. But if we add those confidence intervals in, we can get predicted ranges for different management systems. And then we can plot your value of organic carbon and compare it to the predicted ranges for baseline management, soil health management, and reference sites. And that will give you a sense of how healthy your soil is when it comes to organic carbon. But it's customized to your texture, so your specific clay percent. So in this case, we've got a soil that is doing pretty great in terms of organic carbon. It's actually beating the predictions for baseline and soil health sites. And yet it's still fairly far away from those reference values. We can do this for all of our indicators to give a complete picture of how a producer is doing with these soil health indicator values. And then we deliver a soil health report, which includes a detailed interpretation of the results for both of our both the SHI indicators and the SHAP report. We'll also be providing resources on soil health and cost share programs, but Ryan will get into that more. I want to end today by recapping. So we found that soil health practices did significantly improve all of the soil health indicators that I showed once we controlled for soil texture, for organic carbon, and aggregate stability. So that means that progress is working. You know, the practices that that farmers in the Golden Horseshoe are adopting around minimizing tillage and incorporating carbon, incorporating cover crops, are bringing our soil health values higher and increasing the soil health of our soils. However, there is still room for management innovation to further improve our farmed soils. So if you look at the difference between the blue line and the green line on all of the plots I showed, there's still a lot of innovation space. So today on World Soil Day, I want to offer a call to action. So what practices can we implement to further regenerate our soils? With that, I'd love to hear your questions or ideas, and feel free to email me at jbauer@soilhealthinstitute.org. Thanks. And with that, I want to turn it over to Ryan, who will discuss a little more about our sampling program and next year.
Excellent. Thank you very much, Jenny. Really appreciate that. So hello, everyone. My name is Ryan Carlo. I'm the soil health specialist with the Greenbelt Foundation. I'm going to be talking about our soil health sampling program, and I'm going to be starting off with eligibility for our 2024 sampling. So first, we want to talk a bit about the target fields that we're looking for. So we have two kind of categories in this section. We're looking for fields that are growing corn or soybeans in 2024, or another spring planted grain or oilseed crop. And these can be in any kind of combination of rotations, so corn, soy, wheat, or any combination such, as long as the field we're specifically sampling in 2024 was planted in the springtime. This is to try and keep the reference values for each of the sites that we're sampling consistent. We're also looking for samples from a wide variety of management systems. So across this project, we're looking at sites that are completely no-till, sites that are minimum till or using strip till, and sites that are using more conventional tillage practices. We really want to get a wide variety of management systems in our data set. Second category of sites we're looking for are reference sites. So these are long-term undisturbed fields, looking for sites that haven't been tilled in 10 to 15 years or longer. These can be long-term hay fields, animal pastures, fence rows, orchards, fallow lands, or other types of properties that just have not been tilled in the last couple of years, last 10 at least. So the next part of the eligibility criteria for our program is soil type. Because we're comparing soils to each other, we really want to make sure that we have soils that are consistent enough that they can be compared to each other. So for our program, we're looking at broad categories of this simplified texture triangle that the Soil Health Institute has worked on. This orange section here, which includes sandy loams and loam soils. The green section here, which is sandy clay loams, and some loams as well. And then this blue section of the triangle, which is mostly silt clay loams and silt loams. So those are the ideal soil types for our project. We're looking to avoid soils that have too much clay content or too much sand, again, as they're quite different from the other soil types, and it will make giving a good comparison between all the samples in our project more difficult. Our target locations for next year are the regions of York, Durham, and Niagara. So we're continuing our sampling in the Golden Horseshoe, and these are the last 3 regions that we need to sample for our baseline data. So in York and Durham, these maps here show the orange is suitable soil types. So those 3 categories of soil types that we're looking for are on the orange soils here in these maps. So as you can see through York and Durham, almost all of the farmed land is likely on a very good soil type for us to sample. So anyone farming in York and Durham is very likely to be eligible should they meet the other criteria. In Niagara, it's a bit different story because of the higher content of clay in some of the farmland. So it's going to be a bit trickier, but we do have lots of land in northern Niagara and along the Welland River that we think is likely in that clay loam category. And we're very happy to work with any farmer in the Niagara region to make sure that we find a sample for them, either through our program or through another program. If you have multiple fields, there's very likely one of the fields will have some type of clay loam that we can work with for our project. So the process of our project is quite simple. We understand everyone's very busy, so we're trying to do most of the work on our end, so we're not holding you up with doing your processes. So the first step is a brief phone call in the winter of 2024. That's when we're going to determine eligibility, talk to you about your soil type and what kind of crops you grow, and provide some more background information on the project. Then before the sampling period, we're going to do a quick call, likely after planting, to make sure that we can schedule a day that we can come out and actually do the sampling. So we have a sampling team ourselves who does the sampling. So it takes about 2 hours, and we'll just need to confirm access to the particular field we're sampling with you as well. And again, this will take place in late May to July of 2024. While we're sampling or shortly afterwards, we're going to have a short discussion with you on your field management history. So looking at the last 5 years of that field in terms of fertilizer applications, cropping practices, and tillage practices as well. And then we're going to deliver the results report in the new year following the year that we sampled in. So the information that you receive from this project, so all participants get a really comprehensive soil health report, including the results for the field that we sampled on, but also soil health management information to help with decision making. We're also going to be including resources on some of the factors that influence these soil health indicators and how potentially specific BMPs can help. We're also going to be providing information on locally relevant cost share programs from our project partners, including OSCIA, conservation authorities, and other groups that are providing funding for best management practices in your specific region. And the one thing about this project is because it's a 4-year project, we hope to have continued conversations and support with all our participants so that we can follow up if a new program is developed or we get more information into our database that changes things. We want to be able to continue to provide information about soil health and not stop after the report is delivered. And then again, this is a 4-year project, so our sampling plan for 2025 and 2026 will be sampling grain and oilseed farmers all across the Golden Horseshoe, so it'll be open to everyone from Niagara up to Durham. There are going to still be some eligibility requirements, but again, very happy to work with anyone to determine those eligibilities one-on-one. We're also going to be focused on informing all involved in the agricultural industry on soil health testing and how these results can inform management decisions on your farm. Also hoping to demonstrate BMPs that have the ability to improve soil health on specific fields. So if you or anyone you know farms in the York, Durham, or Niagara areas and you'd like to get involved and get a free soil health test, be happy to have you in the program. So just contact us here at our email is soil@greenbelt.ca. We also have a link to our program here on the Greenbelt website, and this contact information will be provided in the follow-up email as well. So that concludes our talking portion of the webinar. So thanks very much for listening. We have some questions in the chat, but I'm going to start with a quick one, maybe to Jenny, if we want to talk about just why should farmers do a soil health test?
I think that's a great question. One reason to do a soil health test is to get an idea of how healthy your soil currently is, which I think both SHAP and the SHI method do a good job of that? And then, you know, are you thinking about implementing certain practices, or have you done so on certain fields, but not others? It's great to have fields to compare by soil health testing. But then I think also, you know, in terms of like, why should a farmer be thinking about soil health at all? I think there's definitely a lot of economic incentives. You know, we hear that that it can be really important for water management. So, you know, getting some drought resilience and also absorbing floodwaters and so forth. So I think there's some really powerful incentives for thinking about soil health and testing it.
Excellent, thanks. From the Q&A, maybe for Interjot, you can answer this one. Is the score development process similar to the CASH approach from Cornell?
Good question. Yeah, it's yeah, the approach is pretty similar. So what we have been what the Cornell people are doing in terms of comprehensive assessment of soil health, that's what they call it. Like it's called, it's a CASH soil health framework that's developed. I think it was for New York State, but yeah, the approach, I would say it's pretty similar to what the scoring functions they're using over there. Yeah.
Excellent, thanks. Another question here, I can answer this one. Were any of the fields you tested using biosolid fertilizers, or did they disclose any fertilizer practices? So yes, we did sample fields specifically in Halton region, a little bit into Wellington as well, that were using biosolids. Absolutely welcome to have them in the program. And we do have a discussion point with farmers about what types of fertilizers they're using, including any organic amendments, which the biosolids would fall into. Another question here from Nick. Would you be able to do the SHAP sampling at the same time as they're doing soil samplings for the YEN project? Interjot, if you want to jump in on that one.
Sorry, can you explain like what's the, I'm not so familiar with.
I think it's the basic of the question is that the SHAP sampling itself is fairly simple, that you could do it while you're doing other soil health sampling, that it's not, you know, a 3-hour process. I think that
Yeah.
would be
No, I think it's, yeah, it's pretty yeah, like it's similar to what the other soil health sampling programs do. So we ask, as a part of our ground truthing SHAP project, we ask for 2 bags of soil, just to be sure that the lab has enough sample for completing all the analyses on all the soil health indicators that they are measuring. So yeah, so earlier we were asking for 1 bag of soil from each submitter, but now we have increased it to 2, just to be on the safer side. But yeah, the samples can be collected at that point as well when you're doing the assessments for other indicators.
Yeah, and
just real quick, so as far as I'm aware, YEN does their soil testing around March, which is a bit early for some of these soil health indicators. June is kind of the ideal timing. So typically that I think that has lined up in the past with like their later tissue testing timing.
Yeah.
Great. Yeah, thanks, Sebastian.
But the SHAP test is meant to be done with regular cores, 6-inch cores like you would take for soil fertility.
Yeah, yeah, exactly. Up to 15 centimeter, 6-inch depth is what we ask for SHAP.
Yeah, our test is a little, I mean, we collect the 0 to 15. We do 15 to 30 as well for carbon stocks. So we also book density sample from 0 to 15 and 15 to 30, but otherwise similar. And also we collect little aggregates for slakes.
Yeah.
Great. Jenny, I'll ask this question for you. Does the time of year, does it matter when we're doing the sampling for soil health?
I love that question because I actually analyzed it in our data. So we found that, so first of all, we, you know, in our project, we weren't trying to answer that question. So we don't, for example, have data from every like time in the year. But we did see a small but significant effect of dates sampled on our respiration. However, that was kind of an artifact of our sampling. So we sampled our soil health sites mostly in July, whereas the other sites we spread out over June and July. So I would say with the indicators we're working with, keeping it consistent and keeping a fairly narrow window of like 4 to 6 weeks, but I think we kind of stretched it to 8 weeks is important. And then just making sure that you're sampling around the same time each year if you're comparing through time. So maybe, in your own project, you're sampling during a different time of year, but you want to be comparing to a bank of samples if you're doing the scoring or rating that were sampled at similar times. Do you have more to add to that, Indrajot? Most of the SHAP samples are from the early part of the year, correct?
Yeah, but like we have been so I would say yes, that was the initial protocol, but there have been some instances where the growers have gone up to like late fall in terms of submitting their samples. So I definitely would agree to Jenny, like there is definitely a time trend. So especially because most of our soil health indicators that we are measuring as a part of SHAP, they are all mostly labile except organic carbon or organic matter. So you expect a lot of influence of time as that time when you're sampling those or collecting those samples. So yeah, especially if you're building a baseline for your soil health, it's very important to be consistent to the time points that you're sampling each year. And also in terms of comparing your samples to other fields or, from one section of the field to another, I would say time it's a very important variable in that case. Yeah.
Great. I think I can ask one final question here, Jenny, if you can give a short answer to this one, is just why is our project sampling reference sites? What do we find important about getting that information out to farmers?
Right. That's a good question. Well, I think, overall, we want to be able to know, if there's a region where for whatever reason, soil health practice adoption hasn't occurred yet, or maybe it's just starting to scratch the surface. I think it's important to know what the maximum values are for soil health indicators so that we can get a sense of what the full potential of soil health is there. That doesn't mean that we're necessarily going to attain it, but I think farmers have been comparing cropland and fence row soil health for a long time, and it can be a useful indicator to see where you're at on the soil health journey.
Yeah, absolutely. That's great. So,
Any other final questions, Ryan, or anything else you'd like to end off with today?
no, I think we covered it pretty well. So thanks, everyone. Really appreciate it.
Yeah. Thank you, everyone, for joining today and for your thoughtful questions. I encourage you to check out the email that will follow the webinar today. It will have a recording of this session, so you could share it with colleagues or friends or other farmers who might be interested in participating in one of these programs. It will also include links to the programs that we discussed today and the new app, Slaking app that's launching that Jenny covered, and email addresses for the folks on the line if you have any follow-up questions. The email will also contain a link to a survey so you can provide some feedback on the webinar today and add any additional comments you'd like to share. So thank you very much for joining us today. Thank you for all of our speakers for your time and expertise and sharing that with everybody on the line, and for all the work that you do for soil health in Ontario. And with that, happy World Soil Day. Thank you.